So today the topic is soil analysis is the first step for soil fertility management. So you can know that healthy soil, healthy plant, and healthy people. Healthy soil, the foundation of life. A healthy agriculture soil is one that is capable of supporting the production of food and fiber to a level and with a quality sufficient to meet human requirement together with continued delivery of other ecosystem services that are essential for maintenance of the quality of life for humans and the conservation of biodiversity. So on left side, you can see that it is a productive soil. So productive soil gives higher crop production. But you know that when the soil is degraded, then you are getting very low crop production. Then sustainable soil management. Sustainable soil management is essential for getting food, feed, fiber, fuel, medicine, and ecosystem services. So there are two principles. First, everything extracted from the soil, such as carbon and nutrient, must be returned to it. So you know that when we are growing the crops, then plant extract carbon and nutrient. So we have to return to the soil. Second, the soil should not be left without vegetation cover, which has a positive effect on carbon sequestration, means grow cover crop. Then overuse and underuse of fertilizers. Overuse of fertilizer, particularly nitrogen, may lead to nitrogen deposition. Chronic nitrogen addition may increase soil nitrogen mineralization. Excessive use of nitrogen fertilizer leads to soil acidification. A negative soil health trait leads to the imbalance in nutrient availability. An underuse of fertilizer means that soil nutrient exported with crops are not being replenished, leading to soil degradation and declining yield. So you can see that on the right side, on dangerous ground, land degrading is turning soil into desert. So if we are not taking care of the soil and we are applying the fertilizer without carried out the soil analysis, then you can see here the land is degraded. So at present, our soil productivity has been declined due to faulty handling of land creates soil degradation. So you can see that at present we are applying huge quantity of fertilizer, but we are not getting the crop response to the fertilizers. So the main reason that the soil is degraded and we are not apply the balanced nutrition, we are apply the nutrient imbalance in the soil. Then wide nutrient gap between nutrient demand and supply. So you can see here that nutrient removal is greater. So here the blue color, you can see that nutrient removal is greater and nutrient consumption, red color, it is nutrient consumption, and yellow color, you can see that it is nutrient gap. So in 2003, gap is 10, 2010, gap is 12, 13, but at present, gap is more than 22. So projected food grain production relation to nutrient, NPK consumption and removal and gap. So you can see here, that nutrient imbalance lead to soil health deterioration. So here, you know that the ideal ratio is 2:1:1, nitrogen, phosphorus, potassium. But here you can see that when the ratio is not imbalanced, then the production of the rice is low. When the balanced production of the NPK, then you can see that nitrogen 5.5, phosphorus 1, and potassium 2:7. Then we can produce the highest grain yield of rice. So nutrient balance is required for production of the higher grain yield of the rice. Then decline nutrient use efficiency. So you can see here in 1970, the fertilizer use efficiency 13.4, but at 2010, 2005, it is declined to 3.7. So why the nutrient use efficiency decline? Because we have killed the microorganism in soil and soil becomes dead due to excessive use of chemical fertilizer, pesticide and fungicide. Then apply balanced fertilizer. So you can see here that emerging micronutrient decline deficiency due to imbalanced fertilizer use. So you can see that in 1950, we have only nitrogen deficiency. In 1960, we have deficiency of iron and nitrogen. And at present, we have the deficiency of the multinutrient, that is nitrogen, iron, zinc, phosphorus, potassium, sulfur, manganese, molybdenum, and so on. So here you can see it. Then application of balanced fertilizer on the actual limitation of nutrient in the soil is very crucial to increase production and productivity of crop. So you can see that when we carried out the soil analysis, when the nutrient status is high, then we are not getting the more response. We are often getting the response of only 10%. When there is a medium soil fertility status of the nutrient, either major, secondary, or micronutrient, then we are getting the 50% crop response. And when our soil, we are carried out the soil analysis and we saw that when there is a low nutrient status, then we are getting the 90% greater production. So when we have getting the low or medium, then we should have to apply the nutrient as per the recommendation made by the State Agriculture University of the India. Then soil testing is the first step. So this practice of soil sampling and testing will help the farmer to know exactly what nutrients are required by the crop. So soil testing is the first step for soil management. So having in mind the soil type and its nutrient capability will help one to know what kind of crop to grow and kind of pest to use during pest and disease control. You know that we have to feed 9 billion people. So we have to apply the balanced nutrition in the soil, and we have to improve these not only soil fertility, productivity, but soil sustainability. Then why test soil health? So soil health can be tested by chemical test. So in soil testing, we are doing the pH, then major nutrient, nitrogen, phosphorus, potassium, soil organic carbon, and micronutrient. So soil testing is very important. Before sowing cultivation of crop in every season, it is very important to check the soil for its nutrient status. If any nutrient is less deficient or more excessive, toxic than required amount, necessary measure can be taken. Soil testing encourages healthy plant growth and development of a crop by using base fertilizer recommendation. Soil testing categorizes soil saline, alkaline, or mineral soil. Farmer can be advised to convert such soil into normal soil. So you know that problematic soil means saline, alkaline soil, or acidic soil, and the crop productivity of the saline, alkaline, acidic soil is low because the nutrients are present, but the nutrients are not available to the plant. So we are getting the low crop production in saline, alkaline, and acid soil. So by soil testing, farmer will know the health of their soil, whether it is rich, medium, or poor, and according they plan to improve it. So you know that Mr. Narendra Modi sir was the chief minister of the Gujarat. Then he started the soil health card, and it is very important. And he has started the Krushi Mahotsav and launched to other state of the India. Then Justus van Liebig law of minimum. So you know that that scientist has found out that crop yield is proportional to the amount of the most limiting nutrient, whichever nutrient it may be. If the deficient nutrient is supplied, yield may be improved. So you can see here the phosphorus deficiency, here zinc deficiency, and here iron deficiency. So there are 17 essential plant nutrients required for optimal plant growth and development. If one of these nutrients deficient, the crop life cycle will be negatively affected, even when all other essential nutrients are adequately supplied. Suppose zinc is deficient and all the nutrients are sufficient in soil, your crop has been declined. Similarly true for iron and phosphorus also. So here you can see that it is a maize crop, corn crop, zinc deficiency, and on right side, it is a healthy plant of the maize. So the farmer are apply only sufficient NPK fertilizer in soil in maize crop without soil analysis. After 1 month, observe zinc deficiency and harvest only 3000 kg per kg grain yield per hectare. If he would have been carried out soil analysis before sowing of maize crop and applied 25 kg zinc sulfate per hectare, he would got about 4,000 kg per hectare maize grain yield. Resulted in farmer had obtained more income of rupees 20,000, that is 25% more income by correction of zinc deficiency. If they had not carried out the soil analysis, they obtained only 3,000 kg grain yield of the maize. So the cost of the soil sample analysis is only 1,000 to 1,300 one soil sample, and cost of 25 kg zinc sulfate is rupees 2,000. So total cost is rupees 3,300. So net profit of the farmer 16,700 per hectare. That is why soil analysis is very important, and you are getting extra income of 16,700 per kg per hectare in 1 hectare land. Moreover, application of unbalanced fertilizers in soil deteriorate the soil health and soil become degraded. Therefore, need to carry out soil analysis before sowing of crop and apply nutrient as per soil test value. The farmer will definitely obtain not only maximum grain yield and profit, but keep soil healthy for their future generation. Then synergistic reaction. Avoiding antagonistic interaction, increasing synergistic, one can have a positive influence on yield. Then antagonistic reaction with other nutrient, you can know that phosphorus has the antagonist effect with the zinc and iron. So in the interaction of phosphorus and zinc designates as P-induced zinc deficiency. This nutrition disorder in the plant is commonly associated with the high level of available P or with application of P fertilizer to the soil. This deficiency symptom can be corrected by application of zinc deficiency. Means I cannot tell you that suppose your soil is sufficient in phosphorus and you cannot carry out this soil analysis of the phosphorus or soil. Where, again, as per the recommendation of the State Agriculture University, you are apply the phosphorus fertilizer. So you know that the native zinc or iron, or you can apply with the chemical fertilizer, then there will be antagonist effect P and zinc. So you are not only lost the cost of the phosphatic fertilizer, either DAP or single superphosphate, but the zinc and iron native or applied with chemical fertilizer is not available to the plant, and we are in loss. Then type of soil test. So mineral content, pH level, soil moisture, then salinity and structure and soil texture. Then what do you need? For a soil analysis, what is required? So soil auger. Then if the auger is not available, then spade or trowel can be used for taking the soil sample. A bucket for mixing and collecting soil, so it is required. Then clean plastic sampling bag able to hold 1 kg and waterproof marker pen is required. So when to soil sample? Soil sample can be taken at any time of year, but avoid sampling within 2 years of applying lime or within 2 months of applying a compound fertilizer, organic manure, or more than 50 kg per hectare of nitrogen. Then where to sample? So it is important, larger field should ideally be subdivided into 4 hectares, that is 10 acre unit, and sample separately even if they appear uniform. Separate samples should also be taken from distinct area which were manured differently or which have different soil types or topography, even if there is only one crop. Suppose you grow the one crop, suppose maize crop, but topography is different, then you have to take the different crops or different soil samples, sorry. So avoid sampling hotspot areas such as around gates, area where lime or manure has previously been dumped, and areas where livestock gather such as water trough. Field margins should also be avoided as there tend to be variation in fertilizer application in this area. Then sampling objective or requirements, so you have to take the soil sample. A minimum of 10 to 15 cores should be taken from relatively small area, 2 to 4 acres. Taking 20 to 30 cores will provide more accurate result. But you have to take 10 to 15 from 2 to 4 acre, and you have to take the soil sample zigzag method. As you know that a snake is walking, so you have to take the soil sample zigzag method. Then a consistent sampling depth for all crops should be used because pH, organic matter, and nutrient level often change with pH, with the depth. So you should have to keep a uniform depth of the soil. Suppose 6 inch, then you keep 6 inch uniformly. For pH, organic matter, phosphorus, potassium, and zinc estimation. Then sampling objectives. So tree fruit, then we have to take soil sample at 1 feet or 12 inch or 30 to 40 centimeter depth. Then when sampling is specific area, zigzag method I told. Then if balanced fertilizer has been used on the previous crop, then it is suggested that the number of core taken should be increased to minimize the effect of the individual core on the composite sample results and obtain a better estimate of the average fertility for the field. For permanent sowed or long-term no-till field where nitrogen fertilizer has been broadcast on the surface, a 3 to 4 inch sampling depth would be advisable to minor surface of soil pH. Why I told that why we have to take 10 to 15 pores from 2 to 4 acre? Because you know that soil is a heterogeneous mass. Soil is not homogeneous, and therefore we have to take the representative soil sample. Then soil sampling procedure. So you can see here, zigzag method, then make the V shape, then it is given. So it is a use zigzag method and it is V shape, 15 to 20 places, then 20 to 30 centimeter depth. And for a horticulture crop, you have to take 30 to 40 centimeter depth. So make V shape, cut 20 to 30 centimeter deep with the help of spade and take a 1 centimeter slice from the smooth side. So soil sampling procedure for chemical analysis. Drive the auger to a plow depth 20 to 30 centimeter and draw the soil sample. Take a representative soil sample. I told that representative soil sample is very, very important. Then method of soil sampling. So awareness creation on sampling, then collection of soil from pit. So you have to mix it. Then preparation of composite sample. So you mix it well. Then you make 4 parts. Opposite portion you have discarded. Then mix it again. Then make 4 parts. Then opposite portion discarded. We require only 250 to 500 gram sample is sufficient for the major secondary and micronutrient analysis, pH, EC, and organic carbon. Then what test should I ask? So I told that pH, electric conductivity, then major nutrient, secondary nutrient, and micronutrient. Then soil pH and nutrient availability. So I told that acidic soil, so the pH is less than 6.5. When there is alkaline soil, the soil pH is greater than 7.5. But majority of the crops, agriculture crop require the neutral soil pH means 6.5 to 7.5. And at that soil pH, nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, majority of the nutrients are available to the plant. But alkaline soil and acidic soil are fertile, but it is not productive due to high pH of the alkaline soil and low pH of the acidic soil. So nutrients are present in soil, but it is not available to the plant. Then carry out soil and water and plant testing for nutrient as well as plant disease diagnosis. So I am working as a chief soil health specialist in Sowing Agri Clinic and Agri Laboratory, Bhavnagar, Gujarat. So it is our address. So we have ICP, we have scientific instrument. The cost of this instrument is 1 crore rupees, and we are getting the result immediately and accurate results and reliable results. So it is our Sowing Agri Clinic and Laboratory. So you can see here the pH, EC, organic carbon, nitrogen, phosphorus, potassium. It is value and it is a range. Similarly, for secondary nutrient, sulfur, then micronutrient. So all these nutrient I am giving the advice to the farmers on the basis of the soil test value. Then soil management for soil sustainable agriculture. The soil sustain most living organism, being the ultimate source of their mineral nutrient. So what happened at present, the farmers are using farmers are not carried out the soil analysis and they are applying the nutrient or fertilizer as per his or advice of the fertilizer shop, resulted in imbalance of the nutrient and crop productivity has been declined. Good management of soil ensure that mineral elements do not become deficient or toxic to plant and that appropriate mineral element enter the food chain. Soil management is important both direct and indirectly to crop productivity, environment, sustainability, and human health. To achieve future food security, the management of soil in sustainable manner will be challenged through the proper nutrient management, appropriate soil conservation practices. So, how to manage soil fertility? So, you can adopt integrated nutrient management, means you can use organic, inorganic, biofertilizer for higher crop production and for soil sustainability. Then mulching and cover crops. Means don't keep soil naked one, but it should be covered with the crops, cover crops. So there will be no less erosion. Then fertilizer application. So apply the fertilizer on the basis of the soil test value. And it is a well-known fact and proved throughout the world that balanced nutrition you apply in soil, then plant will take balanced nutrition, and there will be better growth and development, and you are able to produce the maximum grain yield, fruit yield, or whatever it may be. Then minimum tillage. So it is proved that when you are carry out the minimum tillage, then less disturbance of the soil microorganism and soil structure also maintained. So we have obtained the higher crop production. Frequent tillage is not advantageous. It is proved by the experiment. Then crop rotation. So it is a proof that multiple crop in rotation break weed, insect, and disease cycle. Rotation produce healthy and productive crops. So you can take the suppose rice, then cotton, then cowpea. So continuous. In a 1 year cycle, you take the legume or pulses or oilseed crop. So that crops fix the atmosphere nitrogen in soil, and we require less chemical fertilizers, and we have maintained the soil fertility. Then fallowing of land. While fallow is commonly referred to as resting period for agricultural land between two cropping cycles, during which soil fertility is restored, it has more role than just fertility restoration. United human being take the rest up to 6 to 8 hours sleeping. Similarly, land is also required the rest in a year, 3 to 4 months. Then it is just for your knowledge. So we are given the low, medium, and high. So you can see that organic carbon, then nitrogen, then phosphorus, low, medium, and I am recommending to the farmers. So we have in English, then we have in Hindi, and we have in Gujarati recommended to the farmers. And if any farmers have problem, then they may contact me, and I will advise. So you see that zinc. So they point less than 0.5 ppm zinc, then it is low, and we should have to apply 25 kg zinc sulfate per hectare. When it is 0.5 to 1, then we should have to apply 8 kg zinc sulfate per hectare. When it is high, more than 1, then if you will find the zinc deficiency in standing crop, then we have to foliar spray 0.5% zinc sulfate, and we should have to make neutral pH with the lime or buffer solution, so there will be no any acidic effect on the leaves. Similarly, for true iron, then sulfur. So sulfur is also important. Then manganese, then copper, then boron. Boron, you know that at present we are observing the boron deficiency, and boron is required for the pollen tube germination and seed size. It is very important. Fertilization. Then molybdenum. So molybdenum deficiency also we are observed at present. But in 1950, we are observed only one deficiency nitrogen. But at present, you know that we are growing the intensive crops continuously throughout the year, and we have the irrigation facility in India more than 50%. So at present, we have the multi-nutrient deficiency, and it is very difficult for the farmer to manage to correct the multi-nutrient deficiency. And therefore, we should have to apply the organic manure, then biofertilizer and chemical fertilizer for maintain the soil fertility and sustainability. So M.S. Swaminathan told that the future belongs to nations who have gains and not guns. So I have completed the presentation. Now we have to switch over any query of the farmers.
Okay, thank you very much, sir. Thank you for an
Okay.
informative presentation. I will take up the questions from the participants. In the meantime, can you please stop sharing the screen, sir? And please on your video so that we can see.
So what I do?
Stop sharing the screen, sir.
Okay, okay. Okay.
And please on your video, sir, so that we
Okay.
can see you.
Okay.
Yes,
Okay.
sir. This is fine. Thank you.
Okay.
I'll move
Okay.
on to the questions. The first question is, based on what you have seen, are many farmers doing soil testing? Why is there so little awareness?
Pardon?
Sir, I repeat the questions.
Okay.
Based on what you have seen, are many farmers doing soil testing? Why is there so little awareness?
Yes, you know that government of India and every state has started the soil health card. But I observe that the farmers are taking the soil sample, but it should not be taken the representative soil, because I told that the soil is a heterogeneous mass. So we have to take the soil sample from 15 to 20 places in 4-acre land and make the representative soil sample and send to the laboratory. Then you are getting the better results.
Okay. How long will it take for a farmer to do a soil test? What is the proper procedure? What is the cost involved?
You know that there are different costs of the soil sampling analysis. But you know that at present we are taking the 1,250 rupees for major nutrient, secondary nutrient, micronutrient, pH, ECN, organic carbon. But we have the latest instrument. I told that the cost of that instrument is rupees 1 crores. But suppose you use the not costly cost, then also some take 500 rupees, 700 rupees, 800 rupees for the soil analysis. So it depends on the instrument and accuracy of the instrument. If costly instrument, then we are getting the reliable results and accurate results. But suppose we have not, it is a cheap instrument, then we are not getting the reliable results.
Okay. What is the approximate cost involved, sir?
Pardon?
Approximate cost involved.
Approximately cost about 1,250 rupees for all the nutrient, major, secondary, micronutrient, pH, ECN, organic carbon. And along with the advice, because you know that suppose we are become sick, then we are going to the laboratory, then they give the laboratory report. But we approach to the doctor, and doctor will give the advice that you have to take this medicine. Similarly, in agriculture also, soil analysis is also important, but advice, what the farmer have to do, that is more important. So I am doing this the business. So advice is also for the laboratories, they are also good, require very important.
Okay. I'll move on to the next question, sir. By doing a soil test, what are the benefits for the farmer?
Okay. You know that I told that our topic is the soil analysis, the first step soil management. You know that I did PhD in soil science and I have 48 experience. But looking to the soil, I can't say that that soil has a deficiency of major secondary nutrient. So by carrying out the soil analysis, we can know that which nutrient is deficient soil, which nutrient is medium in soil, and which nutrient is high in soil. So accordingly, we can plan to add nutrient in soil. So I told that application of balanced nutrition in soil, then we get definitely higher crop production. So we can save the money, because you know that the cost of the fertilizer is increasing very high. So we are apply the balanced nutrition and we are getting the more crop production. Because you know that the input cost is important, because output is the grain production or food production. But we have to minimize the input, and it should be required by scientifically like soil analysis.
Okay. And one of our member, Mr. Mohan Rao, is asking, what is the price of the machine for soil testing?
I told that soil analysis, suppose you use the atomic absorption factor photometer for micronutrient, then at present the cost is 18 to 19 lakh rupees. But suppose you purchase like our laboratory, ICP OES, then the cost is about 1 crore rupees, installation and other charges. So pH EC meter, the cost is about 10 to 15,000, pH meter, EC meter. But then spectrophotometer normal for phosphorus and sulfur, then it requires about 1 lakh rupees. So I think that for the establishment of the laboratory, at least 5 to 10 lakhs rupees is required. And you know that government of India gave this subsidy for the students of the BSc Agriculture honors or other BSc science student also.
Okay. Again, Mr. Mohan Rao is asking, soil test for construction and soil test for agriculture is the same, sir?
No, for a construction purpose, it is different because in construction, you know that construction, suppose a black soil, clay soil, then you know that when you apply the irrigation water to the black soil or clay soil, then it will cracks. So it is important for the engineer to find out the soil texture and structure. Whereas in our agriculture, we have to know all the physical parameter, chemical parameter, and biological parameter also. We have to also look the fungi, bacteria, and nematode in the soil. So then organic water holding capacity is also required. Then what type of soil texture, whether it is sandy soil or sandy loam soil or black soil. So accordingly, farmer can plan the crops on his field.
Okay. The next question is, what does Multimate Agromat Private Limited in Bhavnagar, Gujarat do? What services does it provide farmers?
You know that at the Bhavnagar, we are carried out the soil, water, and plant analysis through ICP OES scientific instrument. And we have to provide the service to the Amazon or Flipkart. So farmer can purchase the Sevama soil testing kit. Then they can send the soil sample, water sample, or plant sample directly to them. And you know that I am working at Anand. So we have the program. So they send the soil, they carried out the soil analysis at Bhavnagar, then they send the soil test report to me, and I will give the advice on English, Gujarati, or Hindi, whatever the language the farmers wants, and I will give the advice to the farmer. And if any farmers have any query, then I will also advise to the farmers.
Okay, I have completed the queries from our participants. Sir. There are many participants from your side also. I see some questions posted in the chat window. One, Mr. Ashok is asking, is there any correlation in between CEC and fertilizer application?
CEC and fertilizer application, yes, there is a correlation. You know that when the cation exchange capacity, you know that black soil or clay soil or medium black soil have higher cation exchange capacity, whereas sandy soil have low cation exchange capacity. So suppose when the cation exchange capacity of the black soil or medium soil, then you can apply, suppose continuously ammonium sulfate, then there is a less problem. But in sandy soil, when you apply the continuous ammonium sulfate or urea fertilizer, then there will be chances to your soil become acidic. Another thing, in a sandy soil, the cation exchange capacity is low, so we have to apply more organic matter because we have to increase the cation exchange capacity. Whereas in a black soil or a clay soil or medium black soil, cation exchange capacity is higher, and it is a inherent fertile soil, so we require less nutrient. as compared to the sandy soil.
Okay, and again, Mr. Ashok has posted a question. When same sample is being tested in two different laboratory, there is significant difference in test values. Why it is so, and what should farmers do?
Actually, what happened, you know that actually soil sampling is equal. Suppose you have taken, the farmer have taken, I told that the procedure they have carried out, I told as similar. Then the farmer get the representative soil sample and make two portions. Suppose send to the one laboratory and another portion sent to the another laboratory. Then there will be no significant differences. But what happened? You know that farmer take the soil sample, suppose sent to the Bhavnagar. After 10 days, 15 days, he draw the another sample and send to the suppose Anand laboratory. So there is difference because you have taken the different time and your different procedure for the soil sampling or depth or location is also changes. The location is not remain same. So there will be changes and you know that there is a human slight error, but it is not significant. Suppose nitrogen is low, then it will not jump to the medium or high. Value slightly change, but it is categorized in low. High categorized, it remain in high. So it is not much significant differences. And you know that accuracy, I learned that soil sampling is more important than chemical analysis. And that is why I told that you have chosen good topic that why soil analysis is important. So soil analysis, soil sampling procedure is very, very important than the chemical analysis. Because farmer will collect the soil sample. So he has to take the representative soil sample and send to the laboratory. And we also provided, our Seva Agri Clinical Agri Laboratory provided the whole procedure in Seva Agri kits also. So I will give the full advice to the farmers and accordingly farmer will take the soil sample and send to us. So
Okay.
farmers are satisfied.
Okay. Again, the next question is from Mr. Ashok. What is your views on mobile soil testing kit? Does it give same result like lab test?
You know that in a micronutrient, ionization is required. The main principle is ionization. When you atoms are ionized, then it gives the characteristic wavelength. Correct? But in mobile, you know that in ICP, OES or atomic absorption spectrophotometer, the temperature about 2000, 3000 degrees centigrade temperature. So proper ionization is taken place in such type of instrument. But I don't think that in a mobile application, you are not getting as accurate result as atomic absorption spectrophotometer or ICP, OES scientific instrument. But you get the idea. But for accuracy, we require the sophisticated instruments like atomic absorption spectrophotometer and ICP, OES scientific instrument and others also.
Okay. Mr. Ashok again is asking, is it necessary to draw soil samples from different depth of soil?
Yes. You know that suppose you have the problem. Suppose you know that you have the alkaline soil. Then alkaline soil, you know that the different profile, O origin, A origin, B origin, C origin. So in alkaline soil, you know that salt has been laid down from O origin to A origin and B origin. So and suppose our crop is not give the response, and suppose you take suppose we want to grow the horticulture crop like mango or other crops, guava, then at that time we have, suppose the fruit production is very low at that time, we have to find out the calcium carbonate content, then sodium content in the soil, then potassium calcium ratio is also important, then sodium bicarbonate is also important. So it is necessary, but when you have the problem, then because in normal course there is no problem. But when the crop productivity or soil is degraded, then we have to carry out soil analysis up to 0, 30 centimeter, 30, 60 and 60, 90. So it is a profile chemical analysis is required. So for the solution, what is the remedy? So accordingly, we can choose. Suppose you know that in a Bhal area near Ahmedabad, they have the soil saucer type topography. So we cannot recommend it to apply the gypsum in such type of soil. So for that, we have to use the soil resistant crop like rice, wheat, gram, sorghum, then barley. Such type of crop we have to choose for the higher crop production.
Okay, and the next question is from Mr. Krishna Sahane is asking how many pH contamination in the soil?
Actually, it is a good question. You know that soil pH is very important. I told that 6.5 to 7.5 pH, then majority of the nutrients are available to the plant. The micronutrients, zinc, Fe, Mn, copper, boron are more available at 6 pH. But molybdenum, micronutrient, more available in alkaline soil. So soil pH is very important. And you know that microorganisms also multiply at 6.5 to 7.5 pH. So when we have the alkaline soil or acidic soil, then their population will be declined. And whatever the nutrients you apply in soil, that is not available to the plant. So in problematic soil, saline, alkaline or acidic soil, we should have to apply 25% more fertilizer than the recommendation dose, because that fertilizer not available to the plant. It is fixed in soil, and therefore we have to add more fertilizer in soil.
Okay, thank you very much, sir. We have now completed the round of questions.
Okay, thank you very
On
much.
behalf of agricultureinformation.com, I like to thank Mr. Praveen Chandra Chaturlal Patel for the talk and for answering the questions. I like to also thank all the participants in the meeting. This meeting will now be closed.
Madam, can
Yes,
you share me the video to me?
Yes, sir. We will do it, sir.
Okay, thank you very much.
Thank you, sir. You are welcome.
Okay.